Because you wouldn't anyhow do this.
All right, so today we are going to be talking about
You said prepare for a quiz Thursday.
oxidative. Why?
There's a quiz Thursday, right?
Oh, great. I think the only reason it's not Tuesday is
I heard.
Speaking of that, plan on a quiz next Wednesday in this class.
Yes.
Unless that really doesn't work for you guys, then I can move it, but
No, it's good.
it's good. I'll put up some Yankee cards this weekend. I actually did it for the vaccines class. So oxidative phosphorylation, this is essentially talking about the electron transport chain. So just sort of as an overview of the electron transport chain.
Since Monday, but I kept forgetting to turn them in.
Recall that the previous steps of aerobic respiration were producing NADH and FADH2. These molecules are currently in their oxidized form or reduced form. They're currently in their reduced form because they're carrying electrons, right? So they're going to be oxidative, and in the process of oxidation, they're going to lead to the phosphorylation of ADP to form ATP. So that is why it is oxidative phosphorylation. This process is going to consume oxygen, and there's really two steps to oxidative phosphorylation. The first step is the electron transport chain, and the second step is ATP synthase. The electron transport chain creates an electrochemical gradient. So what's an electrochemical gradient? Electro refers to charge. Chemical refers to chemicals, molecules, so something that's physical. So we have a charge and a physical substance, and then we have a gradient of those two things. So the mitochondria is a multi-membrane structure. That is Nebraska, no. So we have the matrix on the inside. We have the inner membrane space. This is the inner membrane. This one here is the outer membrane. The electrochemical gradient means that you have chemicals that are different on each side of the membrane, and the charge is different on each side of the membrane. This chemical happens to be a proton. So all these pluses represent H+, meaning that the pH on this side of the membrane is higher or lower than this side that has fewer H+ on it. It'd be lower, right? So you have a charge on this side. This side is more negative. This side is more positive. So this is sometimes referred to as the P side, and this is sometimes referred to as the N side for positive and negative. And then this side has more protons, and this has more or less protons, so less H pluses. So that's what the electrochemical gradient is. The electrochemical gradient is created by the electron transport chain, so the electrons flowing through that drives the electrochemical gradient. And ATP synthase is then a separate step. So you have a high concentration of H+ on this side, low concentration of H+ on this side. What does the H+ want to do? It wants to return, right? It wants to drive back so that it's at equilibrium with equal concentrations on both sides. It does this through ATP synthase, which takes an inorganic phosphate in ATP and synthesizes ATP from it. So that's the overview of oxidative phosphorylation. We're going to first go into details about the electron transport chain and how they are actually functioning, and then come back and talk about ATP synthase, and then a few other things related to that. So the membrane transport proteins. There are four main ones that are described as complexes or by the enzymatic reactions that they catalyze. So complex 1 is also known as Nadh Dehydrogenase. It is responsible for taking NADH, also takes a H+. The H+ is really not going to be that important to remember. And Q, which is quinone, and converts it into NAD+, and ubiquinol, which is QH2. So you reduce this to quinol in this process, and the NADH gets oxidized to NAD+. In complex 2, you have already seen this enzyme. It is called Sustenant Dehydrogenase. Where did we see this? Where did we see sustenate? The Krebs cycle. So the sustenant dehydrogenase is a Krebs cycle enzyme that's actually embedded in the inner membrane of the mitochondria. So it's catalyzing both a Krebs cycle reaction, and then it's directly coupled to the electron transport chain. It was the reaction that produced the FADH2, and so here we're going to have the FADH2 plus the Q produce FAD and QH2. So these QH2s then are going to be used in the next reaction, which is denoted as complex 3 and called cytochrome C Oxido Reductase. So we take the QH2, we have 2 cytochrome C1 in its oxidized form, and we basically take the electrons from the QH2, you reform Q, plus we end up with 2 cytochrome C1 in its reduced form. And then the final complex, complex 4, is Cytochrome Oxidase. Here we have 4 cytochrome C that are reduced, and we have oxygen. We are going to form 4 cytochrome C that are now oxidized, and we have a water molecule. Because of this, we're going to have some H+s. I didn't draw the H+s here, kind of intentionally. I should have done that up here as well, because they are going to be participating in another in something else that these four complexes are undergoing. All right, so how this works, you'll notice that 1 has the electrons flowing from 1 to 3 to 4, and 2 has it flowing from 2 to 3 to 4. So it is not that it goes 1, 2, 3, 4. It goes 1, 3, 4, or 2, 3, 4, depending on whether it came from, the electrons originally come from NADH or FADH2. If we look at the order of molecules, because there are some things that I left out, you can see that the molecules get reduced in this order, starting with NADH or FADH2. So they go from NADH to the ubiquinone Q to cytochrome B to cytochrome C1 to cytochrome C. There's another cytochrome that's cytochrome A1 and A2 together, and ultimately then the electrons go to oxygen. So this is denoting them all in their NADH is in its reduced form. These are all in its oxidized form, as I have denoted them, because they have not received the electrons yet. We know that this is the order because we can use different inhibitors to stop different places in the cycle of it. So, for example, rotenone is an inhibitor of the Nadh dehydrogenase. So here, if you add rotenone, it stops this. You end up with NADH building up, and then all of these remain in their oxidized forms. Similarly, you can do that for every step along the way. Another example is antimyosin A, which blocks that step of it, and then inhibitors of cytochrome oxidase include cyanide and carbon monoxide, and you can stop it that way. And so you guys probably are familiar with cyanide and carbon monoxide as poisons. They poison you because they essentially stop the electron transport chain from running and would kill you because you can't make ATP anymore. Questions thus far? There are alternative ways that electrons can move into the electron transport chain. We'll talk about one of these. Well, we've talked about one of them already, and we'll talk about another one of them later. One is through glycerol 3-phosphate dehydrogenase. So we actually kind of did talk about this one when we talked about the catabolism of glycerol. This is going to take the glycerol molecule and reduce it to dihydroxyacetone phosphate. In the process of this, it takes the FADH2 and transfers its electrons to ubiquinone to form ubiquinol. And so you have the FADH2 coming in there and producing it that way. Similarly, you have another reaction that looks like this with the fatty acid CoA dehydrogenase that functioned in the breakdown of fatty acids. It's going to take FADH2 and form QH2 and enter essentially like the Sustenant Dehydrogenase would, directly contributing it into this part of the cycle. All right. How much ATP per NADH molecule do you get? 2.5. How much ATP per FADH2? 1.5. Looking at this, you notice that both of them form QH2 and then move on to the next step in the electron transport chain. So you may be wondering, hey, why is there a difference? Because they're kind of producing the same thing along the way, right? Are you wondering that?
Yes.
Since I pointed it out and told you to wonder about it? All right. The reason for this is because of how the electron carriers are ultimately using the energy from these electrons. So as we are moving along in this reaction, the matrix here, and this is the inner membrane space, and then we have our different complexes along the way. As the electrons move through there, these complexes are going to be transporting protons to the inner membrane space. And the number of protons that are moved are going to differ depending on the complex. So this is complex 1, this is complex 2, this is complex 3, and this is George. You don't have to laugh at my dumb jokes. I won't like you anyway.
Okay. Maybe I'll laugh harder at them. Please don't. My eardrum hurts.
Wow. Please do not laugh. It hurts me in my ears and in my calf, says Dr. Seuss. Becca?
It didn't even rhyme.
This is not Dr. Seuss Mecca.
Oh, yeah. Wow. Every time you do that, it throws me off. I'm like, where is he going?
Where is he going? We do not know. Where is he going? Hey, Avery, want to see my toe?
His face of disgust is what really sold that.
All right. So back to the electron transport chain. Recall in the first reaction that we had, where did it go?
I do not know.
Thank you. Recall that I showed that you were actually using a proton in the reaction. So this is going to consume 5 protons, but it only displaces 4 protons to the membrane, because one of these is actually going to be used to form the ubiquinol from ubiquinone. So that's why you don't necessarily have an equal number on both of these sides from this. This reaction here is going to not actually consume anything. So you, from FADH2 to QH2, the hydrogens are moving from the H2, the FADH2 to the QH2, so there's no change there. And it's also not transporting electrons here through it. So this is part of the reason why NADH is making 2.5 ATPs while FADH2 only makes 1.5. It's because you don't get these protons moved from the membrane when you go through FADH2. Complex 3 then is going to take 2 protons from the matrix, and ultimately there will be 4 protons produced in the inner membrane space, And here you're going to have 8 protons on the matrix and 4 protons on this. I'm not going to expect you to remember the numbers. The big key here is recognizing that complex 1, 3, and 4 are transporting protons across the membrane. 2 is not transporting protons across the membrane, and that is why you're getting more ATP from NADH than you are getting from FADH2. It's because you have more protons being transported across the membrane in this situation.
Why is it turning 2 hydrogens to 4?
Because some of the protons are produced in this case during the reaction, just like one of these protons were used during the reaction.
Okay.
If you recall, this is taking the QH2 and forming Q. Those protons don't go into the cytochrome C that's getting reduced, so you have 2 protons being produced from the reduction, the oxidation of the QH2 to quinol. All right, so we have a high concentration of protons on the inner membrane space and a low concentration in the matrix. This is now going to drive ATP synthase. So I drew this backwards. Yes, I'm aware of that.
Didn't think you'd hear me say that.
So the protons flow through here, and as they do, they're going to take ADP and an inorganic phosphate and make ATP. So this is a phenomenon that is caused by the electrochemical gradient. If the electrochemical gradient was neutral, or so that there wasn't a gradient, or if it was reversed, you would actually have the reaction moving the opposite way, because the breakdown of ATP to ADP and PI is exergonic, and so that would actually release energy and drive protons the opposite direction. But because you have a high concentration of protons on this side of the membrane versus that side of the membrane, you drive it so that it's towards ATP synthase. The force that the electrochemical gradient drives is known as the proton motive force. And this model of how ATP is produced from the proton gradient is known as the chemi-osmotic model. So those are not really all the same thing, but kind of referring to the same phenomenon. They're different components to the phenomenon. The model uses the electron gradient to drive the proton motive force to make ATP from it. Ways that we know that this is how it occurs, if you would take the mitochondria and artificially decrease the pH on the inner membrane space, what have you essentially done? Decreasing the pH does what? It increases the concentration of H+. So what is the purpose of the electron transport chain? to increase the hydrogen concentration on the inner membrane space, right? So if you would artificially add pH or add protons here and decrease the pH here, you've essentially done the work of the electron transport chain, right? If you do this, or conversely, if you increase the pH here, ATP synthase starts independently of the electron transport chain. So you can artificially create ATP just by changing the pH on the sides of the membrane of the molecule. Other ways that we know that this is how things work. So you can perform an experiment where we're going to look at the oxygen consumed. So if we look at the oxygen consumed, we start here adding an inorganic phosphate and ADP. So that's what happens here. On this side of the membrane we have time. No O2 is being consumed yet because we haven't produced or haven't provided the cell any energy, or the mitochondria any energy for this reaction. So this is an isolated mitochondria. If we add succinate, so succinate is an intermediate of the electron transport chain, is going to allow ATP to be produced, or allow the Krebs cycle to run, and therefore will allow the electron carriers to produce. You start to see the oxygen consumption go up. And then if we add cyanide, cyanide was what? What?
Poisonous.
Poisonous. What was it inhibiting? The cytochrome C oxidase, the last step of the electron transport chain. Suddenly, the O2 consumed is going to flatten out. That should make sense, right? You inhibit the enzyme that is using the sorry, got something stuck in the back of my throat. You inhibit the enzyme that's using oxygen, and you're not going to be consuming any more oxygen. If we look at the ATP synthesized during the course of this reaction, it is going to follow right along. So you add this, you're not going to synthesize any ATP until you add some sort of thing that can give electrons to the electron transport chain. And then as that happens, the ATP synthesized is going to go up. And then if you add cyanide, you no longer can provide electrons. You can no longer create the proton motive force. So if you stop oxygen from being used, the electron transport chain does not run, and therefore you don't have the proton motive force being driven, and you're not having ATP synthesis. All right. Let's look at this again. But we're going to add some different molecules this time. So we'll start off with the O2 consumed. All right. We add succinate. Nothing happens. Why? Because we don't have any ADP and inorganic phosphate, right? So let's add those in here. Now what's going to happen?
And go up.
It's going to go up. All right. Now let's say, so this is O2. Let's make sure that we note that it's the consumption of O2. Let's say that we add oligomycin. Oligomycin is an ATP synthase inhibitor. What do you think that's going to do to the oxygen that's being consumed? It'll flatten it. Why do you say that?
Because ATP won't be synthesized anymore.
Okay, so if ATP isn't going to be synthesized, why isn't ATP being synthesized?
Because the oligomycin is an ATP synthase inhibitor.
Okay, and if we inhibit the ATP synthase inhibitor, protons are no longer flowing through the gradient. So now we have a buildup of protons on one side of the membrane versus the other. When you have a high concentration of that happening, you can eventually make it so that it's too difficult to move protons across the gradient. And now the electron transport chain is going to stop because the gradient is too great for the energy to be produced here. So we add the oligomycin and this levels off. All right, ATP synthesized. So we add succinate. What's going to happen? Nothing. We add ADP and an inorganic phosphate. It's going to start going up. We add the oligomycin. It remains flat. All right. Here, we add dinitrophenol. So DNP, this was historically used as a weight loss drug. It's no longer used as that because one of the side effects was death.
Of course.
So they pulled it off the market. How it worked was that it basically was called an uncoupler. So it uncoupled the oxygen consumption from ATP synthesis. What it does is allow protons to move across the inner membrane without the use of ATP synthase. All right, so you add DNP, what's going to happen to the ATP being synthesized? What's that? Are we going to start making ATP? No. So it's going to remain flat, right? What about the oxygen consumption? Why did the oxygen consumption stop before?
The electron transport chain shut down.
Why did the electron transport chain shut down?
Because the concentration of hydrogen was too high.
Okay, so what's the DNP going to do to that?
It'll reset the gradient so that you can continue the pump of the hydrogens.
So what's going to happen to the O2 consumption? It'll go up. So now the electron transport chain is functioning independently of ATP synthesis. This is why it was called an uncoupler. You uncoupled the consumption of ATP, or the consumption of oxygen, from the synthesis of ATP. No, that's not good, unless you're trying to lose weight. And apparently that's still not good, because you might die as a side effect. No big deal.
Is that the same guy that was tested in like the UK? Is it that one?
I'm not sure.
Yeah.
One of the side effects of DNP was elevated body temperature.
Yeah, it was.
Why will this result in elevated body temperature?
Because the energy of the electron transport chain is no longer going into ATP synthase, so you're just giving it off in heat.
Very good. So thermodynamics, energy is not created nor destroyed. So the energy has to go somewhere. It's no longer forming the gradient, right? So you don't have the potential energy there. The kinetic energy is moving it down the concentration gradient. Usually the kinetic energy was used to drive the ATP synthase and ATP synthesis, but with no ATP synthesis occurring, the energy has to be lost as heat, because it can't just dissipate and become nothing. You know, thermodynamic laws and such, physics. Sometimes your body will actually do this intentionally. So you have a protein known as thermogenin. Thermogenin is also known as uncoupling protein. I'm not sure what the abbreviation. Uncoupling protein 1. And it's essentially a channel in the mitochondria that allow protons to flow through it in the absence of ATP synthesis. And this causes elevated heat. Anybody know where you find thermogenin?
Would it be in your immune system?
Nope.
Babies.
What's that?
Babies.
Yes, actually. Babies have high levels of brown fat. Brown fat is brown because it contains mitochondria. And the mitochondria then are there not to produce ATP, but to keep the babies warm. There is also evidence that populations that live in more cold climates, like the Eskimos, have more of this than you guys probably do. I'm unsure if that's a evolutionary response where they have evolved to have more of the ATP, of the uncoupling protein, the thermogenin, present, or if it's an adaptive response where your body signals to produce more of it. It could be a little bit of both potentially, because they're living in an environment it's going to be more advantageous to have more of this to stay warm, and so they have, naturally higher levels of it. But your body can also signal, hey, you've been cold all this time, let's upregulate the amount of this protein so that we can keep you warm. So it's not necessarily two independent mechanisms for keeping this up. But in babies, they need to stay warm, so the thermogenin, if you look at the brown fat in babies, they have a lot more of it than you guys do. You do have some brown fat, but at one time it was thought that you lost all your brown fat as adults, but you do have some present, just not very much compared to all the white adipose fat that you guys, well, some of you guys know, that you have in your body. All right. So Wednesday, we'll have a quiz. It will be over fatty acid metabolism plus the stuff we talk about today. On Monday, we're going to be talking about some shuttles. So you have NADH in your cytosol. It needs to get to the mitochondria. It can't freely pass through the mitochondria. The NADH from your cytosol, how did you produce that? In glycolysis. So we're going to talk about how glycolysis gets the NADH into your cells. And then we will discuss the regulation of this process. So this is the quiz. This is what you get to look forward to on Monday. All right, have a good day.
It's the retro warm-up from like the 80s. It's the one Andy wore it at the national tournament, and he got on Twitter and stuff. And I'm like,